called binary fission.
Okay.
When it's prokaryotes. It's called mitosis when it's eukaryotes.
What's the difference?
There's not much difference. I mean, it's a lot faster. It's a single chromosome dividing instead of a number of chromosomes. They don't line up in the middle and do all that.
So does that the prokaryotic cell doesn't like get smaller as it just doubles its size and then cuts in half?
It's kind of it's kind of stretching is what it's doing while it divides. So I don't know. It's just a different name, really. It's really basically the same thing as cell copying itself. I mean, the steps are a little bit different. Okay, so now we go on to the next. The next chapter is chapter 10, which is a different type of cell reproduction. It's the type of reproduction done by sex cells to allow you to make a baby. We're talking about sex now. He's so cute. And the baby too.
Of all things, you could have gotten a baby.
The baby's like, oh my god. You see his face? It's like, oh no.
Okay, let's think about what has to be done to make a baby. To make a baby, you take two individuals, male and a female, in human. And the man gives some of his traits to the baby, and the woman gives some of her traits to the baby. And the way this is done is the man makes sperm cells, and the female makes egg cells, and a sperm has to come together with an egg. And so what ends up happening is there's a special form of cell reproduction to make these sperm and egg cells. They're called gametes. Is that distracting y'all? The sperm and egg cells are called gametes. And we have to make these gametes in a special way. And that way isn't called mitosis. It's called meiosis.
Wait, both the sperm and egg cells together are...
They're called gametes. Gametes are sex cells. And the sex cells have to be made by this process. And it's a little bit different than mitosis. What we're going to have to do is we're going to have to cut the number of chromosomes in half. So if a normal human cell has 46 chromosomes, which it does, a normal human cell, almost all the cells in your body have 46 chromosomes. But your sex cells only have half of that. A sperm cell, which kind of looks like that, has 23. And an egg cell, which is from the female, has 23. And when the sperm cell combines with the egg cell, after the parents have sex, 23 plus 23 is?
46.
So that's the normal number. That's the right number. So somehow you have to make cells, gametes, that have half the number of chromosomes. And I'm going to show you the process by which that happens because it's an important process in lab.
And that process is meiosis.
Meiosis. Do it, Sam.
So meiosis is the process that halves the number of chromosomes.
That's it. Now, I know one of y'all noticed this. I don't know if anyone else did. Do you notice that I have a green set and a white set of chromosomes up here? Cole saw that. Cole, you should be a detective when you grow up.
Sex cell. I don't know.
What? You okay, Emery? All right. The sex cell detective? chromosomes from its paternal side, from its father's side, and got the Y chromosomes from the mother's side. Each parent donates chromosomes to the new cell, you see. And you got half your chromosomes from your dad and half your chromosomes from your mom. And that's why there's two sets of colors. Question?
So when someone says like, oh, she is more like her mom and her, that's not really true.
Well, it can be true because of the way the chromosomes are expressed. We're going to talk about that later. You might express more of your mom's chromosomes than your dad's. They're not equally expressed. But yeah, it's not true though. You do get half your chromosomes from your dad and half the chromosomes from your mom. And if you were to take a cell that's going through mitosis and take a picture of it during prophase, which a computer can do, it can just flash a picture here. And then the computer can cut out the chromosomes and put them together by size. Put those together, and those together, and those together. We call that type of picture a karyotype. And they're showing one right here. This is a karyotype. And it shows all 46 chromosomes. They call these two pair 1, these two pair 2, pair 3, and all the way to pair 22, so that's 44 chromosomes. And then those are the sex chromosomes that we're going to talk about later. This is a female. This cell is from a female person because there's 2 X chromosomes. We call those the X chromosomes.
Isn't when one of your X chromosomes is messed up? Isn't that cause like Down syndrome?
No, it's actually an extra chromosome number 21 causes Down syndrome. When you have 3 of them instead of 2.
What if you have one of them?
If you have one of them, you got big problems and you won't live.
When my mom was pregnant, they told her I was going to have Down.
Wait, Emery had her hand.
Okay, so karyotype is just one of like a karyotype just puts.
A karyotype is when you take a picture of one of your cells during prophase, and the computer puts the chromosomes together, pairs them up into what we call homologous pairs, and then lines them all up on a chart. And so we call these chromosomes that are alike homologous. That's the word you need to know. Homologous. Chromosomes that are alike. The only difference being you got one from your mom and one from your dad. But they carry the same information. So for instance, you might have a gene right here for blue eyes from your mom and a gene right here for brown eyes from your dad. If you get a blue-eye gene from your mom and a brown-eye gene from your dad, you're going to end up with brown eyes because brown is dominant over blue. We talk about that later too.
Wait, how does the gene pair up when somebody doesn't have one of the same? I don't get it.
Say that again?
Like when. Oh, okay, so never mind.
Okay.
Wait, why don't we have certain colored eyes? Like, why doesn't anyone have an orange?
They only come in a couple varieties. We're going to talk about that later. Can you wait 2 chapters?
Oh.
I'm sorry. It's pretty complicated. And I don't want to get into that yet because we talk about it later. So let's look at the process by which we form sex cells. It's called meiosis, and it occurs in steps. And I have these steps. Hold on. I'm skipping. Oh, the dogs. Okay, I have these. We'll get back to all that. It occurs in steps. And this is how it works. And this is actually not from your book. This is from an older version. But I wanted to show you this because I think it's the best way to teach it. So here we have a cell, a normal cell. Although it's not located in your arms or on your face or in your muscles, this cell is located in your gonads. That's your reproductive organ. Let's say this is in the testes of a man. We're looking at a cell inside a man's testicles. This is a lot bigger than it would actually be, you understand. That was also a joke. Now this cell, listen, this cell is going to end up making 4 sperm cells. The way it divides, it's going to make 4 new cells. Okay? So how does that happen? Well, it goes through the same phases that we studied before. Prophase, metaphase, anaphase, telophase. But it goes through them twice. So we're going to do one division, which we call Meiosis One. And then we're going to do a second division, which we call Meiosis 2. And this is going to split our cell up twice. The first division is Meiosis 1, and a second division is Meiosis 2. And we're going to end up with 4 cells with half the number of chromosomes. Remember, normal mitosis is just a single split of 2 cells that are identical copies of one another with the normal number of chromosomes. We're going to split into 4 cells, so we have half the number of chromosomes.
So the first time they split, they each have 46, and the second time they'll all have 23.
You got it. You like that?
I don't understand. If they split, then why wouldn't they copy themselves again? To have double.
Because we want to produce sperm and eggs that have half the number of chromosomes. We don't want a normal cell this time.
So why wouldn't they just have the first time and just have Meiosis One?
Well, because when we start during the S phase, we're starting by copying the DNA exactly. Remember, all these chromosomes during interphase are sitting there, laying around in the nucleus like pieces of string. And remember, during S phase of interphase, the DNA copies itself, makes exact copies. So let me put those copies up here. Copy, 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 copy. Now we have exact copies. And now we're going to start this process of meiosis, which basically evolved from mitosis. It's similar to mitosis. And so, just like in mitosis, the nuclear envelope dissolves, and the little centrioles move to opposite sides, and they shoot out spindle fibers. Shoot out spindle fibers. And the chromosomes attach to the spindle fibers. Only this time they do something a little bit different. They're going to find their homologous pair and get right up next to it.
What stage is different?
This is all called Prophase One. It's similar to mitosis. The only difference being that these chromosomes find their homologous pair and pair up with it.
Is it just randomly in the cell, or are they lined up in the cell?
No, they're randomly in the cell.
So this is not like displayed in a book anywhere?
Yeah, they basically show it on the bottom of 173.
Do they go through prometaphase?
Yeah, I mean, prometaphase is where the chromosomes attach to the spindle, so yes. But I'm not going to really worry about it.
And they don't go through interphase, right?
Yes, we already did interphase. That's when we copied the chromosomes. Interphase was before all this.
Do anything coil up?
Yeah, the chromosomes coiled up. Now we can see them. Then they go find, they attach to the spindle fibers and find their partner. Everything's the same as in mitosis, except for they find their homologous pair.
What does it mean by like their pair? What does that mean?
Like they're same pieces.
You
The
see
same
how
sizes?
these two are the same size?
Uh-huh.
They had to find one another, you see. They had to seek one another out. We didn't see that in the last time we did normal mitosis. They didn't do that.
Oh.
But here they do.
Okay.
That's the difference. Yes?
How did they get in the cell together if they're from two different people?
When the sperm fertilized the egg.
Yeah, but does it just create one giant cell?
Yes.
What happens?
When the sperm fertilizes the egg.
Yeah.
Mhm. The sperm is real small, by the way. Doesn't way smaller than the egg. Hundreds of times smaller. Okay. So now what happens? Well, an interesting thing happens at this part of meiosis. It's called crossing over. The chromosomes actually kind of lay on top of one another. And the chromosomes will switch pieces like this. And we call this crossing over. And it doesn't just happen in one spot like I showed you. It happens in many spots all over the chromosome. Crossing over of pieces, switching of pieces.
Is this for Prophase One?
Prophase One.
Why are they switching over pieces?
That's a great question.
To create variety.
It creates variation is what it does. It makes pieces, it makes chromosomes that have never existed before. Because it switched some genes around. Remember how I said that the dad could have given a brown-eyed gene and the mom could have had a blue-eyed gene? Well, here, it switches them. Now this chromosome might have a brown-eyed gene and this chromosome has the blue-eyed gene. And that's never existed before. This chromosome has never existed before because it was different when it was in your dad. So basically, in your gonads, you shuffle the genes around. This is like shuffling up the deck of cards. You shuffle the genes around and you make chromosomes that have never existed before.
And this is all happening during Prophase One.
Prophase One.
It says crossing over. Oh wait, never mind.
Crossing over happens during Prophase One. So imagine that pieces have been switched. And it usually happens about 4 times per sister chromatid. There's about 4 switches of material. And these are switching too, because these things are actually laying on top of one another. So both sides, both sister chromatids switch pieces.
They call this meiosis.
Meiosis. This whole process is called meiosis.
That's Prophase One.
Let me go back here and show you this crossing over.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during Prophase One meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the...
He's talking a little bit above your head here.
Yeah.
So it's a tetrad.
But you can see here, see those pieces switch?
Oh, yeah.
Ooh, that's fun to do.
Oh, so this is like after they're fertilized?
No, this is before they fertilize.
So how is...
How's the this
You
happening?
haven't even got
How does this happen?
yet.
We hadn't gotten to the sex yet.
That's what I thought.
This is still in the
Well, from the
gonads.
parents of
Let's speed
the.
things up then.
From the previous parent that made you. See, the green ones came. If this were one of my cells, the green ones came from my dad, and the white ones came from my mom.
Is it like that in all cells or just your
All cells.
Exactly, that's what I'm saying. So how are we not talking about that? Like, how would there?
I'm talking about
What? Oh, so you had me right. Then I have my dad and my mom. That's what we're talking about.
every cell inside you has half chromosomes from your dad and half chromosomes from your mom.
I get it.
Oh, okay, I get it. So you transfer
Alright.
half the chromosomes of your dad and your mom.
So technically you're a fourth of your grandparents.
So yeah, so we're a fourth of our grandparents.
Yeah.
And
I
then
look
they
exactly like my grandma.
Cool.
I don't look like anyone else like my grandma.
We'll talk about the expression of genes later. Your grandma's genes are more expressed.
I'd rather not.
We'll talk about that later. Can you wait?
No.
Well, you're going to have to wait. All right. Now, let's continue. The rest of Meiosis 1, which is the first half of meiosis, is kind of the same as mitosis. Oh, and by the way, someone's asking what a tetrad is. That's a tetrad.
So the pairs are tetrad?
Yeah, a homologous pair is also called a tetrad. It has another name too. It's called a bivalent. Why does it have three names? Homologous pair, tetrad, bivalent. Some things just have three names. I'm Mr. Willis, Tom Willis, and DJ Grandmaster T. You can call me any one of those. And I'll answer. I'll tell that joke about 12 times before they
A tetrad and what?
A tetrad, a bivalent, a homologous pair is the same thing. So in Metaphase One, which is the next phase, the chromosomes kind of move to the middle. The pairs move to the middle. Can y'all count how many chromosomes this cell has right now?
9.
1, 2, 3, 4, 5, 6. This cell has 6 chromosomes. But a real human cell would have 46.
So we haven't split yet.
We haven't split yet.
So there's theirs and 40 others.
Yeah, there would be these and 40 others, but I can't fit all that on my board. But you're right, in a human,
Need a bigger board.
you would have 43 more, or I'm sorry, 40 more chromosomes. Okay, so, continue. Anaphase One is when the chromosomes separate and move to opposite sides. But it's a little bit different than in mitosis because the individual chromosome doesn't separate in the middle. The homologous pairs separate and move to opposite sides.
What hooks them together?
I don't know.
Centrosomes. They're just next to each other.
Yeah, they're next to each other. I don't know how they're held there. It's a good question.
Well, I don't get the difference.
Well, before it was like this.
Oh, I get it.
In mitosis, they were all lined up like this. And each one split like that.
Mm-hmm.
But in meiosis, the pairs are together. And the pair splits apart. You see what I'm saying?
Mm-hmm.
Dante, are you with me?
Yeah.
Now, I want you to notice something. There's something that scientists have called independent assortment. Meaning, the pairs can split up any way they want to. It's random. In other words, these two can go like this, where the green one goes this way and the white one goes this way. Or they can go like this, where the white one goes this way and the green one goes this way. And the way the next pair separates has nothing to do with the way this pair separates. So if this pair goes white-green, this pair doesn't have to go white-green. It can go green-white. In other words, they they separate independently of one another. And that has a name. It's called independent assortment.
And that's just part of Anaphase One.
Independent assortment happens during Anaphase One. In Anaphase One, the chromosomes separate, and they do it independently of one another. So the paternal chromosomes from the dad can go separate one way, and the maternal can go the other way, or they can go in opposite directions. And each pair separates independently from the next pair. That's called independent assortment. Yes?
How are there like dad ones and mom ones if they haven't combined yet?
Well, because all these green ones, they came from my dad.
And this is you?
And all these white ones, they came from my mom. And then I'm going to shuffle those chromosomes up inside my gonads. I'm sorry I'm talking about my gonads with y'all. And we've got it on tape, so I may be fired tomorrow. You see what I'm saying? If this is one of my cells, each one of my cells has my dad's, half the chromosomes are from my dad, and half the chromosomes are from my mom. But when I'm making my sex cells, I'm going to shuffle them up.
Okay. Got it.
So my sex cells, some of my sex cells might get a lot of my dad's chromosomes, and some of my sex cells might get a lot of my mom's chromosomes.
Okay.
But it's random how they split up. Does that make sense?
May I please go to the restroom?
Yes.
So they don't split up evenly?
They don't have to split up where there's either white or green or white. It's random. That's called independent assortment. And it leads to variation. There's no way you're ever going to get two of the same kids unless they're identical twins. But that's a different process. But there's no way you can ever have two separate kids and have them turn out exactly the same. Because of these crossing over that we do and independent assortment that we do, the kids will always turn out different.
So there's no way.
No way. I mean, the odds are, it's mathematically possible, but you're talking about 1 in hundreds of trillions. That the shuffling turns out exactly the same way.
But it could happen though, right?
It's possible.
Has it ever happened before?
No.
Wait,
Yes.
What is that? I just got lost. What is the
We're
purpose
still a chance.
Someday.
What is it? Someday.
It adds variation.
So it's just making a number for like, so there's more numbers.
Every sperm cell that a man makes is different due to the shuffling that goes on during
So
meiosis.
just making a whole bunch of different ones to increase the probability of.
To increase the variation in a population.
Okay.
Yes.
Okay.
So, the... Okay.
It's alright.
I'll let Isaac go and you think. Yeah.
So this variation is what Charles Darwin was talking about, variation.
Yes.
Okay. If, like, how would it be different if, like, I know, like, the crossing over and stuff would be different because there wouldn't be, like, exactly the same amount of, like, mom and dad genes.
Yeah.
But, like, if the one mom gene went to one side and the dad gene went to the other, then both sides would end up being the same.
No, because what's going to happen is over here, we're going to make a couple of sperm cells on this side, and we're going to make a couple of sperm cells on this side. Okay? If this sperm cell ends up fertilizing an egg, well, that kid's going to have some of my dad's genes.
Okay, I get it.
If this sperm cell ends up fertilizing an egg, well, then the kid's going to have more of my mom's genes. Because the white ones ended up over here, and the green ones ended up over
How do you
here.
figure out what you want?
But if these got switched around and did like that, then this sperm cell is going to have some of my dad's and a little bit of my mom's.
But more your dad.
But more of my dad's.
So
And see, every time you make sperm cells, the shuffling is different. So that all the sperms are going to be different.
Does this happen with, like, animals?
It happens with every organism.
Like, some of the animal species look exactly the same almost.
And now's the time for the cute dog video. Because this is why you shuffle things up. The crossing over and independent assortment shuffle things up, and you might be wondering, why would you want to shuffle things up? And so that's what I have the cute dog video for. To show why, whoops, not him.
What?
Oh. Look at the dog. Look at that one. He's curious. Let's see why. If you notice, none of them are exactly like their parents, and that's from the shuffling that's done. And here's the reason. Here's why you shuffle things up. Pay good attention here. Pay good attention here. And I'm not going to finish this process. I'll finish it tomorrow. But you'll have to read about it.
When any organism reproduces, it makes new individuals that have the same basic characteristics as itself. Offspring share with their parents a kind of body plan that remains essentially the same generation after generation. For example, this mother and her kittens have fur, 4 legs, whiskers, and other characteristics that distinguish them as cats, not fish or birds or people. But each kitten is also different from its parents and its brothers and sisters. Most noticeably, each has different colored fur, but there are many other differences as well. While it is important for all members of any species to have the same basic characteristics, genetic diversity is also important. In fact, genetic diversity helps a species survive.
To see how, let's imagine a mythical species of animals, which we'll call wimpogs. Suppose all wimpogs are genetically alike in every respect, and that they all have characteristics that help them survive in a warm environment. Up to now, wimpogs seem to do fine just the way they are. But now, suppose that conditions change, and the environment grows colder. The same genetic characteristics that help this species survive in a warm environment might now threaten its survival. The species could become extinct. But with genetic diversity, the outcome might be different. Let's suppose now that some individuals of the wimpog species have characteristics that would help them if the climate gets colder. If such a change in climate occurs, these individuals will be better able to survive. If they reproduce, they also will ensure that wimpogs survive as a species.
While wimpogs are imaginary, in reality, different species benefit from genetic diversity. Genetic diversity provides options to a species that may help it survive when conditions change.
So, I didn't get to the end of this. But think about it. If my dad was real hairy, and my mom was not very hairy, and they got married and had me, well, I get my dad's genes and I get my mom's genes, and when I'm making sperms for my kids, I shuffle them up, you see. And it might happen where sometimes when I make sperm cells, they all end up with my dad's genes. But more often than not, when they separate, you'll have some sperm with many of your dads, but some sperm will have many of your moms, and so some of my kids will end up pretty hairy or pretty non-hairy because they're getting more of my mom's genes. But there's so much shuffling that goes on, it just shuffles things up, and usually they end up a combination. But when I end up having kids, I might have some kids a little bit more hairier than others. And if I was a wild animal in the wild and it got real cold, then the hairy kids would do well and the non-hairy kids would all die out. But some of the kids would survive no matter what the temperature was. You see what I'm saying?
Yeah.
So this is, I'm saying this quickly within one generation, but what this shuffling up does is leads to diversity, and diversity helps a population. So we see a lot of the little changes happen here in meiosis when you're making the gametes. Now, I only got through Anaphase One. You actually, in the reading tonight, it takes you through the entire meiosis, which I'll finish with you tomorrow. So you need to read through, read 10.1 and 10.2 tonight, and I'll finish